Good afternoon, everybody. Today's subject is translation, the synthesis of protein from mRNA, and in particular how viruses do it differently from cells. So the start of this is messenger RNA, shown in the upper right. This is a typical eukaryotic messenger RNA. It has a cap at the 5' end, which is very important for translation. I think most of you have learned about this in some other course. This is the structure of a 5' cap. It's a base inverted. It's attached to the second base by a 5' to 5' linkage rather than the usual 5' to 3' linkage. So this confers a number of important properties on the message. It's essential for translation. There are usually 5' and 3' untranslated regions. There's an open reading frame that makes a protein, encodes a protein, and that's defined by an initiation codon, an AUG, and termination codon. Then there's usually a poly A tail at the 3' end. This cap, as we'll see, is important in a variety of steps, not only in translation, but you'll see in processing pathways as well, turnover of mRNA. So that's a typical eukaryotic messenger RNA. That's what we're going to talk about today, mainly. So these 5' end translated regions have important regulatory roles. They can be long, and they often have secondary structures in them. Remember the stem loops and the tertiary kissing loops and pseudonodes. Those can all be in 5' UTRs. And they can inhibit the passage of ribosomes, which have to get through this region to find the AUG. So the secondary structure of the 5' UTR can affect translation efficiency. 3' untranslated region can also affect efficiency of translation. It seems counterintuitive, but the current model of translation is that the 3' end of the message actually circularizes with the 5' end. So that 3' UTR plays a role in translation efficiency, and the poly A tail in particular is necessary for efficient translation. The translational machinery is very extensive. It consists of ribosomes, which are shown here. Eukaryotic ribosomes have two different subunits, a large and a small subunit. And ribosomes, of course, are made of protein plus RNA, and it's the RNA that plays a catalytic role in assembly of the protein. The other important component are the tRNAs, which help to decode the mRNA, of course. These are three different views of tRNAs. A linear view showing the sequence of bases. This is a highly base paired structure, a lot of secondary structure obvious. The anticodon region pairs with the mRNA, and the amino acid is attached at the other end. This is a three-dimensional model, and these are space-filling models as well. These are particularly instructive. You can see how the amino acid is at one end and the anticodon is at the other. In addition to ribosomes and tRNAs, we need initiation proteins, elongation and termination proteins. I'm not going to go into these at all today, but I'm going to tell you the abbreviations that I use so you know what I'm talking about. EIF, EF, and ERF. The little e because these are eukaryotic and to distinguish them from the prokaryotic counterparts. So I want to talk briefly about initiation as it occurs in an uninfected eukaryotic cell, because then we're going to see how a virus modifies this. There are two main ways that you can get initiation of translation, or as we say here, two ways by which ribosomes can bind. The first is a 5' dependent initiation mechanism, which is done by most mRNAs. In this situation, the initiation complex, which we'll define in a moment, binds to the 5' end of the mRNA via the cap, and it moves down the mRNA until it encounters the right AUG, which may or may not be the first AUG it encounters. Sometimes there are others in between. So there's a scanning that goes on here from the 5' end to 3' direction until it reaches the initiation codon. The other mechanism, which was discovered in virus-infected cells, but which now clearly occurs in normal uninfected cells, is internal ribosome entry. And in this case, the initiation complex binds relatively close to the initiation codon. And we'll talk about how that works in a moment. So two main ways of initiation in eukaryotic cells. The 5' end-dependent mechanism is outlined here. It starts with a free 40S subunit, which then interacts with a number of initiation proteins. It binds what's called the ternary complex, which contains the initiator tRNA. That's the methionine tRNA, which is going to start laying down protein at the initiation codon, which includes methionine. And then this binds further proteins, including EIF4F, which we'll talk about. And then finally, this very large complex finds a messenger RNA and binds the 5' cap. So there are a series of protein-protein interactions going on here that localize this to the cap. So first of all, there's EIF4E, which specifically binds the cap structure. So if you took purified EIF4E and a cap and mixed them, it would bind with high affinity. EIF4E in turn binds EIF4G, the red protein. That binds EIF3. And then finally, EIF3 binds the 40S subunit. So a very complicated way of getting the 40S subunit to the 5' end of the mRNA. So in eukaryotic cells, uninfected cells, all of these interactions are needed. Let's take a look at EIF4G, the red protein, because it's going to come back several times as we talk about what viruses do to the translation mechanism. It's the linear diagram of the protein is shown on top. It's quite big. It's over 200,000 Daltons in size. And you can see different domains on this protein that interact with different proteins. So for example, we have the EIF4E binding site here, and that EIF4E is the cap binding protein. We have the EIF3 binding site, and that's EIF3, which is the final linkage of 4G to the ribosomal subunit, and then some binding sites for other initiation proteins, which we can talk about. You also see there's a cleavage site for proteases. And these are viral proteases that cleave this protein as a way of shutting down host translation. We'll get to that later on. So this is 5' end-dependent initiation. This whole complex of proteins basically tells the ribosome that there's a cap here and that this is possibly a good mRNA to translate. So once this complex binds at the 5' end, it then has to get to the AUG initiation codon. And this is a difficult problem because sometimes there's a lot of secondary structure in the 5' UTR. And if there is, the complex has a way to get around it. It has a protein called EIF4A here attached to it. This is an RNA helicase. So as this complex is moving down the 5' UTR, this RNA helicase unwinds the secondary structure, such as this, so that it can get past it. Now, you can put experimentally very, very big, stable secondary structures in this region, and the helicase can't deal with that. So there is a limit above which the helicase cannot get by. And we think this is a way of regulating the expression of proteins. If you have a relatively unstructured 5' UTR, the ribosome can get through very easily. You can make a lot of protein. If you have a lot of structure, it's more of an impediment. You make less protein. This complex at some point scans down the RNA, and this is an energy-dependent reaction, mainly because the helicase needs ATP. And then when it finds the AUG codon, which is shown up on the top here, when it finds it, then a series of reactions occurs. A number of initiation proteins are shed, which aren't needed anymore, including EIF3, because now the 40S subunit is nicely bound to the RNA. The 60S subunit comes in, and now we have an initiation complex ready to translate. And that tRNA MET, which was brought in way at the beginning, the first initiation complex, is still sitting there. And the next thing that's going to happen is tRNA will be put in the next site, the A site, the addition site, and protein synthesis begins. Now, we think that this whole thing actually occurs on circular RNAs. There's a lot of evidence which I won't go into. But in particular, there is a binding site at the N-terminus of EIF4G for a cell protein called PABP, poly-A binding protein. So that protein not only binds 4G, but it binds the 3' poly-A. And a number of experiments in yeast, in vitro, and a variety of systems have provided evidence that this kind of interaction is needed for optimal translation. Nobody's ever seen this in a cell actually happening, so there are some skeptics, but there is a good amount of evidence for this. Now, in virus-infected cells, there are other ways of decoding RNA, and that's what I want to tell you about now. So what we just reviewed was the fundamental mechanism of initiation. That is the 5' end-dependent mechanism of initiation. But viruses have revealed other ways to decode genes. One example is shown here. It's called ribosome shunting. And this is an experiment where we put we take a 5' non-coding region, which is shown in green at the top, and we put it in front of a open reading frame that will produce a protein that we can pick up very easily on a gel by polyacrylamide gel electrophoresis, which is shown here. So we have this mRNA, 5' UTR, has a cap that has an open reading frame. If you place this into cells, it can be translated, and you get these proteins made. Okay, so this is the wild-type construct, if you will. It works pretty well. This happens to be a very structured region, and so it is actually surprising that it is translated. It's translated because the ribosome, it has this particular structure that led ribosomes bypass it, which is called ribosome shunting. The idea is that the ribosomes start to scan in from the cap. They encounter a structure of some sort, and they basically bypass all of that secondary structure. They don't scan anymore, and they go right to the AUG. Now you can see if you put a very stable stem loop structure, which is shown in red here, near the AUG, that has no effect on translation of the protein. And this is interpreted to mean that the ribosome can bypass all of this structure, including this, to land at the AUG. But if you put this secondary structure at the other end of the 5' UTR, right here in red on the bottom, then that blocks translation. So the idea is that the ribosome has to scan a bit, has to enter this complex region before it can bypass. So this is called ribosome shunting. It's been found in a number of virus-infected cells. And the mechanism, or a model at least for the mechanism, is shown here. The idea is that the ribosomes bind the cap, they start to scan, and then they encounter a structure which has the property of telling the ribosome that you don't have to scan anymore, but you can direct to the AUG. So I'm drawing it like this. The ribosome is kind of floating on top of these stem loops that eventually makes it to the AUG, as opposed to having to scan through this region and melt all this secondary structure. So this has been found in a couple of virus-infected cells, some plant pararetroviruses, cauliflower mosaic virus, which we talked about the other day, which encodes reverse transcriptase. They undertake shunting. Some adenovirus mRNAs undergo shunting, and also Sendai virus, a virus related to measles. It's a paramyxovirus. And why do viruses do this? Well, if you do this, you don't need to melt out the structure in the region, because you can jump over it. And that is good because a number of viruses, as you'll see, destroy EIF4F, which is the complex at the 5' end that includes the helicase. So there's not going to be much of this helicase binding to the 5' end. So if these viral RNAs can be translated without that, they're better off. And that's probably why this has evolved. Another one is internal initiation, which I alluded to earlier. So this is a mechanism discovered in virus-infected cells. First discovered in the genome of poliovirus. So here is the poliovirus genome, which is a plus-strand RNA. It encodes a very long protein, a polyprotein we call it, which is processed by proteases, viral proteases. But the interesting thing about this messenger RNA is that at the 5' end, it has a very long untranslated region. When this was sequenced in the 1980s, it's the longest 5' untranslated region that had been discovered. It's about 740 bases. Cellular mRNAs, it's usually 50 to 100 bases. There are some longer ones, but most of them are shorter. So that was puzzling, that it was so long. Plus, in this region, there are a lot of AUG codons before the one that is actually translated. So this is the initiator AUG here, but upstream there are many other codons. And it wasn't understood at all how this could be translated by ribosomes in an efficient manner. Finally, there's no cap on this mRNA. The 5' end of poliovirus RNA has a small protein linked to it. Remember, that's the primer that is used by the polymerase to copy RNA. So with all these oddities, something was up, and sure enough, a group in Montreal did the experiment to show that these RNAs are translated differently, and they're translated by internal initiation. So he made 2 different plasmids which encode mRNAs that could be expressed in cells. So here, these are on the top, the structure of the mRNAs. So they're both capped and have a poly A at the 3' end, and they encode 2 open reading frames. Here's the first one, and the protein is called TK. It doesn't really matter. The second open reading frame encodes a second protein called CAT. And in the 2 versions, the first version on the left, the 2 open reading frames are separated by a random sequence of no import. And on the second, they are separated by the 5' untranslated region from poliovirus RNA. So what happens is in a eukaryotic cell, messenger RNAs typically do not encode more than 1 protein. They are monocistronic. So in a setup such as this on the upper left, where we have 2 open reading frames on a single message, the first open reading frame is translated pretty well. The second is not. And that's because after translating the first ORF, the ribosomes generally fall off and they don't scan down to the second one. A few do, so you get a little bit of protein made, but nowhere near as much as the first protein, the first open reading frame. Now, what this group did was to take the second mRNA with the 5' UTR polio, and they said, how is this translated? And in fact, both open reading frames are translated very efficiently. So they reasoned this must be because this sequence at the 5' end of polio RNA must allow for ribosomes to bind internally without having to scan. So in other words, ribosome doesn't have to scan through here. They can bind right in this sequence. So they called it the IRES, internal ribosome entry site. They did one other experiment to cinch this notion of internal initiation, because the critics were saying, well, you know, without an iris on the left, you know, a few ribosomes can scan through and get some of the second orphan made. Maybe all this iris does is let the ribosomes stay on and scan through more efficiently. So it's hard to argue against that. So they did a second experiment where they took these 2 mRNAs and they put them in cells that had been previously infected with poliovirus. As we'll see a bit later, what polio does in an infected cell is it blocks all initiation at the 5' end of the mRNA. So the cell mRNAs cannot be translated. It has a specific block to capped mRNA translation. So in cells infected with polio, this first RNA on the left, lower left here now, made no protein whatsoever because the ribosomes can't get on the 5' end. So they can't make the first protein or the second. And if we go to the second construct with the iris, the first protein is not made because the ribosomes cannot bind to the cap, but plenty of the second protein is made because the ribosomes can bind internally. They're not dependent on a cap to get into the coding sequence. Right, so that is internal ribosome entry discovered with polio and since found in many other viruses. Now, one of the predictions of this internal ribosome entry site is that you don't need a free end on the mRNA for translation. So cap-dependent or 5' end-dependent translation requires a free end, because the cap has to be accessible, but not an iris. And so someone did an experiment, a very nice experiment to test that. They made circular mRNAs. So a technique was developed to be able to circularize an mRNA. On the left is an mRNA, so there's a coding region here. It starts with an AUG and the terminator, and then there's either an iris sequence or no iris. They took both of these RNAs and put them in cells, and they found that without an iris, the circular RNA is not translated, which you would expect because you need a free 5' end. But with an iris, the ribosomes can bind to the iris even in a circular mode. So this proves that internal ribosome entry doesn't require require a 3 5 prime end. This is what an iris looks like. This on the upper left is the poliovirus iris, which is the first studied. And you can see it's highly folded. This is the first base of the viral RNA. The iris begins a bit downstream from the first base, and this whole shaded region is the iris. And then there's about 100 nucleotides from the iris to the AUG. So what happens is the ribosome actually binds to the secondary structure and then scans to the AUG. These are some irises of other viruses, just to show you how different they are. This is another coronavirus, encephalomyocarditis virus in B. In C is the iris of hepatitis C virus, flavivirus, a different family. And D is the iris of another coronavirus, cricket paralysis virus. This is poliovirus crickets. It's also a coronavirus. But there is no sequence conservation among any of these irises. The only way you can tell if a sequence is an iris is to do an assay like the one we just described, to make a bicistronic RNA, we call it, with 2 open reading frames, put the iris in between and ask if both can be translated. Right? So there's no consensus sequence of an iris. And the genome, our genomes probably have many irises within them. There's good evidence that that's the case. But we can't tell by looking at the sequence, because no one is willing to do the experiment to check every one of them out. This is an interesting iris, the hep C iris, because it can bind directly to the 40S subunit. So this in green is the hep C iris, and the folded area of that iris can bind directly to the 40S subunit without any other initiation proteins. It doesn't even need EIF3. EIF3 is required for the ADS to bind eventually, but the 40S will bind the RNA by itself. So it's probably the most ancient iris. It doesn't need any other proteins except the 40S subunit. So how do these work? Well, they probably work in different ways. Remember, cap-dependent translation depends on getting the 40S subunit to the mRNA by a series of protein-protein interactions involving the cap. Some irises, we think, involve binding EIF4G to the highly structured iris, shown here, and then EIF3 recruits the 40S subunit. So we think this is how the polio iris works. And the significance of this broken tip here will be obvious in a bit. And then the most simple is the hep C iris, in which the 40S subunit binds directly to the structured RNA. So in some way, this structured RNA has evolved to allow initiation proteins or even ribosomes to interact with it. So since the corona irises were discovered, they've been found in a lot of other viral genomes, and here's just a list here, just to show you that they're all over the place, different virus families. And they've also been found in cellular mRNAs. And these are some cellular genes which have been shown to have an iris based on making a bicistronic RNA and showing activity. We don't actually know, yes?
I was going to ask if they were compatible genomes.
This is a good question. There's no evidence that they are. So I don't know if viral irises were first or if the cellular ones came first. We don't even know if these are functional in the genome as an iris, to be honest. So these are all at the 5' end of mRNAs in the cell, and not in bicistronic contexts, as far as we know. And there's a bit of controversy over whether they're actually used as an iris at any time, but there's a lot of ongoing work on this issue. So when we initiate translation, you remember you have the initiator met tRNA sitting in the P site, and then the next tRNA will come in. I want to tell you a very interesting way that viruses get around this requirement for having a tRNA here in the P site at initiation. And this is called methionine-independent initiation. And I'm showing you 2 different mechanisms here, with a plant virus shown at the bottom and an insect virus shown at the top. And basically what is happening in both cases is that the viral RNA is mimicking a tRNA, and it's sitting in the P site of the ribosome, so the ribosome thinks there's a met tRNA in there, and initiation can begin. So here at the top is the situation that happens in cricket paralysis virus. So the viral RNA folds here to look like a tRNA. So the ribosome thinks it's ready to initiate. And here, this is a plant viral RNA in green. The 3' end of the plant viral RNA folds into a tRNA-like structure. And it sits here in the P site. And again, the ribosome thinks everything is fine, and it begins to translate. So this is methionine-independent initiation. So the first amino acid is not a methionine for these proteins. So this is the RNA genome of cricket paralysis virus, which we've mentioned a few times. This was a virus discovered, I believe, in Australia. A farmer went outside one day and saw a field of paralyzed crickets. And he called up a virologist, and they made an extract of these crickets, and eventually found that there was a virus paralyzing them, and it was very related to polio. And this is what the genome looks like. It's very much like polio, except the open reading frame is divided into 2 pieces. There's an iris up here at the 5' UTR, just like polio, and there's a second iris between the 2 open reading frames. And this iris is the one that I've been talking about. Here's the folding of it here. And this is the iris that can fold to mimic the tRNA. So this virus has evolved a way to get 2 proteins from a single message, right? So here you get proteins from here and proteins from here. Normally, if you just had one open reading frame, you'd have to find other ways to get more than one protein from that ORF in a eukaryotic mRNA. Because ribosomes, remember, can only bind, for the most part, at the first initiator, translate and stop, and that's it. They can't reinitiate. So this is one way to extend the coding capacity, if you will, of a viral genome. So I'm going to go through a number of examples of this maximizing coding capacity to show you the different ways that viruses have done that. These are all the mechanisms we're going to go through. Polyprotein, maternal initiation, we've talked about leaky scanning, reinitiation of translation, suppression of termination, and ribosomal frame shifting. And these are some of the viral families that do these. So let's start with the polyprotein. The problem here, again, is that eukaryotic mRNAs are more or less monocistronic. Eukaryotic mRNA, 1 protein, and that's it. 1 open reading frame, 1 protein. And that's in contrast to bacterial or archaeal mRNAs, which are polycistronic. They can encode several proteins because there are signals for the ribosome to bind and initiate within the mRNA. There are no such signals within eukaryotic mRNAs. So viruses have to get around this limitation because they have small genomes, and not all of them can make multiple mRNAs, right? Cells make lots of mRNAs that each encode a protein, but not all viruses are able to do that. So one strategy is to make a polyprotein. So poliovirus, the coronavirus is on top, and the flavivirus is shown on the bottom. They are plus-strand RNA viruses. They make a long protein. And then what do? You have 1 protein, but you need maybe 10 or 12 to make more viruses. They chop the protein up using proteases that are encoded in the polyprotein. So here, for example, polio genome is translated into a very long protein, about 250,000 Daltons. And then within that polyprotein, there are 2 proteinases. One is called 3C, and one is called 2A. And those can chop up the protein, both from within it and externally in trans. And flaviviruses do the same thing. They make a long polyprotein, and then they go one further than polio. They use viral proteases to make some of these cleavages, but they also use host proteases, in this case the signal peptidase, which is normally cleaving the signal sequence off the 5' end of proteins destined for export. And this virus uses it to cleave polyprotein. All right, so that's one way to get more than 1 protein out of an mRNA in a eukaryotic cell. The cricket paralysis strategy is another. You simply put an iris in between 2 open reading frames, so now you can get at least 2 proteins, but in fact, each of these is processed further as a polyprotein, so you can get even more protein than that. As far as we know, this strategy isn't used in mammalian cells or eukaryotic cells of any kind. That's interesting, because maybe because mammalian genomes are so big, we don't have to worry about this, but irises are present, and you would think that they would be used for this kind of strategy. Now here's the third strategy discovered in virus-infected cells, leaky scanning. So remember, the ribosome hits the 5' end, starts to scan and finds the AUG. Well, sometimes it goes beyond it. So here's an example of an mRNA from Sendai virus. This is a paramyxovirus related to measles. And this mRNA encodes, let's see, 1, 2, 3, 4, 5, 6, 7, 8 different proteins from one mRNA. And I'll tell you how a few of these are made. We've already actually talked about some of them. The V protein is made by RNA editing. Remember, in some viral infected cells, the polymerase can add extra bases that are not templated, and that shifts the reading frame, and you can get different proteins. But what I want to talk about here is leaky scanning. Let's focus in on these C proteins, which are the blue ones, down at the bottom of this slide. So the ribosome binds the 5' cap, starts to scan. First thing it encounters is an ACG in position 81. Now this is not an AUG, but it so happens that sometimes ACGs will work as initiators because they're misread by the initiator tRNA. So this protein C' can be made by initiation there. But this is not very efficient, and most of the ribosomes bypass this ACG. A few will translate and be fooled, but most will move on. And what do they encounter next? They encounter this AUG at 104. That's an initiation codon. But it turns out it's not a very good one, because what is around the initiation codon, what we call the context, also affects initiation frequency. So this AUG is in a bad context. So it's translated at a low level. But again, most of the ribosomes move on. And then finally, they encounter this AUG at 114. And that's an AUG, and it's in a good context. So this makes a lot of this C protein. So 3 different proteins. C' and C are the same open reading frame, so their N-termini differ. But P protein is in a different open reading frame altogether, so it's a totally different protein. And then finally, these 2 downstream are translated by ribosome shunting. They're very far away from these AUG codons. It turns out that the ribosomes leap from near the cap all the way down here to translate these proteins. So 3 different examples of decoding on this one mRNA of Sendai virus. So that's leaky scanning, when ribosomes translate a little bit of an AUG or an ACG and then move on. There are other examples of reinitiation. These are all discovered in virus-infected cells. We had no idea that these took place. We figured in a eukaryotic cell, 1 mRNA, 1 protein, but we've learned a lot by studying virus-infected cells. So these are 2 examples of reinitiation. On the top, these are examples from herpes virus-infected cells. These viruses encode mRNAs with an open reading frame encoding 1 protein. But upstream in the so-called untranslated region, there are actually small open reading frames that are also translated. So what we think happens is the ribosomes scan down, they translate this little open reading frame, and then they keep scanning. They don't fall off, because this is a very short region, perhaps, and it allows initiation again, reinitiation. So that's one way it can happen. The second on the bottom is a mRNA of influenza virus. Remember, this is a negative strand virus with 8 RNA segments. And this segment encodes 2 proteins by reinitiation. So the first is encoded by translation at an AUG near the 5' end. It encodes the M1 protein. The ribosomes reach this translation termination codon right here, UAA. And then they go 1 base forward, and they're at an AUG of the second open reading frame. So they make this protein first, they terminate, and immediately begin synthesis of the second, which is in a second open reading frame. So it's a totally different protein. So 2 very clever ways of making 2 proteins from getting around, again, this limitation of eukaryotic cells. Another way to make more proteins is by suppression of termination. So normally, on an mRNA, as the ribosomes reach the end of the protein, they encounter a termination codon. So here's a ribosome which is making a polypeptide chain, and the next codon is a terminator. This termination site is recognized by termination factors, ERF1 and 3. They fit in here. And here they are shown, ERF1 and 3. Their structure looks very much like a tRNA. And they fit into the termination site, and they stop polypeptide synthesis and cause release of the polypeptide chain. But sometimes these terminators are recognized by a real tRNA with an amino acid on that. And that's called suppression of termination. Here's an example, 2 examples in 2 different viral genomes. Here on the left is a retroviral genome. I don't know if you remember the other day I told you that the GAG and PAL proteins are synthesized often from a single messenger RNA. Now in this kind of retrovirus that's illustrated here, the GAG and PAL are separated by a termination codon. Here's the UAG at the beginning of this stem loop structure. So the ribosomes make the GAG protein and then they stop. No more downstream is translated. But PAL is essential. This encodes the reverse transcriptase, the RNase H, and the integrase. So it has to be made. And about 5 to 10% of the time that the GAG is made, ribosomes insert a suppressor tRNA here and make a fusion protein of GAG and PAL. So you get a GAG-PAL fusion, which is subsequently processed by proteases. So at a low efficiency, you get translational suppression to make a longer protein, and it's then processed. And it's not made very much, but it's an enzyme, so you don't need very much of it. So that's retroviruses. On the right is an example from an alpha virus. Here, these viruses actually have plus-strand RNA genomes. Ribosomes can translate it as soon as it gets into cells, and they make this first protein called P123. There is a termination codon in the genome shown by this box, this purple box. But there is still some protein encoded past the terminator. And the virus needs that because it is the RNA polymerase for the virus. So again, about 10% of the time that the ribosomes are coming down here, a suppression event occurs, and you get a fusion protein made, which can then be processed. Now, what causes suppression to happen in these instances? Because it doesn't happen on every messenger RNA. Well, here in the retrovirus example, there turns out to be a pseudodot just downstream of the terminator. And we think that the ribosomes bump into this pseudodot, and that disrupts the whole termination process and allows time for terminators to come in. Now, what gets inserted at the termination codon can either be a special suppressor tRNA. So these are tRNAs that recognize the termination codons exactly. And they encode odd amino acids. So here, there is a suppressor tRNA that will recognize UGA, the terminator UGA, and they put selenocysteine, a very rare amino acid. So there are some rare suppressor tRNAs, but most of the time, the reading or the suppression of the stop codon happens when normal tRNAs misread it. There's a misaligned base pairing between a normal tRNA and a termination codon, and an amino acid gets inserted. So they make mistakes. Another example of expanding the coding is ribosomal frame shifting. This also happens in retroviruses. Now, remember, the GAG-PAL example I showed you, a suppressor tRNA is needed to read through the termination codon between GAG and PAL. For other retroviruses, the GAG and PAL are in 2 reading frames. So there's no way to suppress termination of GAG in this case to get the PAL synthesis. What happens is the ribosomes shift reading frames during translation. So again, most of the time the GAG protein is made, but maybe 5 to 10% of the time this GAG-PAL fusion is made, which can then be cleaved to give rise to reverse transcriptase, and that is made by ribosomal frame shifting. So this is really amazing, and it turns out to happen normally, not just in virus-infected cells. Here's a model just to show you what's going on. On the top is the alignment of 2 tRNAs in a ribosome reading the messenger RNA. So here on the top we have a growing polypeptide chain, and the next tRNA about to go in is a leucine. What happens in frame shifting is that we think the tRNAs slip back one base. It's called -1 slippage. And so now, so originally you have this nice base pairing, UA, UA, AU, etc. It slips back one. There's some mismatches, but there's still some base pairing. So you have a UA here, a UA here. This is not good, the AA, but apparently it's good enough to give translation, because now what happens is the next codon is red, which is in the -1 reading frame. The next codon is going to be AUA instead of UAG, and you now make a fusion protein of a different frame. So that's ribosomal frame shifting, discovered in virus-infected cells, also happens in E. coli, in uninfected eukaryotic cells. So those are 6 ways that viruses can make more proteins with the limitation of this eukaryotic system.
A lot of those ways seem like they just happen by chance accidentally. Does the virus have any way of controlling how those things happen?
Yeah, that's a good question. So these are errors of eukaryotic cells. And in some cases, there's evidence that viruses make proteins that increase the error. So in reading these small open reading frames to the 5' end of the message, and then reinitiation, there's evidence that in some cases viral proteins can increase the frequency. And for some of these other processes, the same as well. So yes, the viruses can help themselves by making gene products that increase the frequency of these methods. All right, the last thing we're going to talk about is how viruses regulate translation in an infected cell. Now, for the most part, when viruses infect a cell, they would like to have the entire machinery at their disposal. They want all the translation apparatus, they want polymerases, cofactors, everything. So they really would like to shut down what the cell does. And one of the prime targets is translation. They shut down translation. So all the amino acids, ribosomes, tRNAs, everything is at their disposal to maximize virus production. So I want to tell you some examples of how viruses do this. And keep in mind that in most cases, we've actually learned how things normally work in the cell by studying how viruses interfere with these processes. So everything I told you about EIF4G and binding to the cap, all that was discovered in part because viruses disrupt those activities. So they're incredible probes for understanding cell function. And a really key regulatory step in translation is at the initiation step. And that's outlined in this picture. So if you remember, ribosomes bind, in cap-dependent initiation, ribosomes bind the cap and they slide down to the AUG molecule. At the initiation step, the tRNA with MET remains, and this protein, EIF2, which brought it in, is released. And that release coincides with energy hydrolysis. So GTP is hydrolyzed at the release step, and the EIF2 bound to GDP comes off. Now, we need to have more EIF2 GTP in order to do multiple rounds of translation. So the cell has developed a recycling scheme to do that. So what it does, it takes the used GDP EIF2, it binds it to a protein called EIF2B, and then it replaces the GDP with the GTP molecule. So this is basically a guanine exchange factor, right? So now we have new recycled EIF2 GTP. It can take up another initiator tRNA and participate in translation. So it turns out this is a very sensitive step for regulation, because if a little bit of EIF2 is phosphorylated, and there are cellular kinases that do this, then when this GDP EIF2 binds EIF2B, it stops there. It cannot exchange the GDP for GTP. So the phosphorylation apparently makes EIF2 bind so tightly to the EIF2B that it can't recycle off the GDP. So the consequence is that when EIF2 is phosphorylated, you inhibit translation. So it turns out that in a number of virus-infected cells, this is a mechanism for shutting down translation. And there are 3 kinases, protein kinases that we know about that phosphorylate EIF2. I would just talk about two of them, PKR and PERK. And they all have a very similar kinase domain, but they also have another domain which senses different things in the cells. So let's start with PKR. This stands for protein kinase double-stranded RNA activated. And this has a domain at the N-terminus that recognizes double-stranded RNA. Double-stranded RNA is typically not found in an uninfected cell. It's usually made when viruses infect the cell. Most obvious is when an RNA virus infects the cell. During the replication cycle, the double-stranded RNA is made. So that activates this kinase. Now, when you infect a cell with a virus, those of you who have taken Moshewitz's course will recognize this, and we'll talk about this later on. The cells can sense the presence of the virus, typically by sensing its nucleic acid. And by sensing it, the cell then turns on the production of interferons, which are antiviral proteins. These interferons then go on and induce the synthesis of other cellular genes which have antiviral activity. We'll talk about this in a subsequent session. One of the genes turned on by interferon is PKR. PKR then goes on to shut down translation. So the idea is, shut down translation, maybe we can stop virus proliferation and save the organism. So you sacrifice a few cells, but the rest of the organism is saved. So the way PKR works is that by itself the protein is not active. So PKR is shown at the top here. Sorry, this is actually another active version. This is inactive PKR here. The two blue molecules with double-stranded RNA binding domains attached to them. This is inactive. It is not a protein kinase as it's made in the cell. It has to be activated by double-stranded RNA. So when two or more PKR molecules bind double-stranded RNA, they are activated. And activated means that they phosphorylate one another. So the two PKRs phosphorylate each other, and once they are phosphorylated, they can then go on and phosphorylate EIF2, the alpha subunit. And that shuts down translation by the mechanism we just talked about. So again, the RNA is usually produced in virus-infected cells. So this is a sensor for RNA produced by viruses and turns off post-translation. So PKR is induced by interferon, and it's activated by virus infection. It leads to inhibition of post-translation. It also leads to programmed cell death, apoptosis. So it's another way the cell is trying to clear the infection. It sacrifices itself, the cell dies, and maybe that will stop the virus from spreading. Now, with any good defensive maneuver of the cell, there is a counter of the virus. And you will see this over and over now as we go through the immune response. Whatever the cell does, the virus counters it. So here I'm going to tell you about how viruses counter this PKR activation pathway. And almost every virus has a way of inactivating the PKR pathway. And that tells us that this is very important for virus defense. Now I'll show you just two examples. One is adenovirus. So adenovirus, early in infection, produces double-stranded RNAs. Now this is a DNA virus. So double-stranded RNAs are made when you have transcription from both strands, and these transcripts hybridize and make a double-stranded RNA. That activates PKR. But what the virus also does, it makes another double-stranded RNA called VA-RNA1. This has the unique property of binding PKR, but it can only bind one molecule of PKR. If you remember, in order to be activated, you have to have two or more molecules of PKR bound to the same double-stranded RNA so they can phosphorylate one another. So PKR binds one VA RNA and can't be activated. So that blocks the activation, no phosphorylation of EIF2 alpha, and protein synthesis continues. So a very clever way. It makes this RNA, which has no other function as far as we can tell, except to antagonize PKR. Another example, vaccinia virus encodes two proteins. Here's one K3L, the second is M156R. Great names, right? And these two proteins structurally mimic EIF2 alpha. So what they do, the virus makes a lot of these proteins. They bind PKR. So PKR there's not enough PKR around to phosphorylate the eIF2 alpha because it's all bound to these mimics. So we call these pseudo-substrates. The virus makes them in excess, sops up all the PKR, and prevents eIF2 from being phosphorylated. And there are many examples of this, which are listed in this table, and they act at every step. So these are all acting in this pathway to eIF2 alpha phosphorylation. And we have here the adenovirus example that we just talked about, VA1 RNA. We have the vaccinia virus, pseudosubstrates, and then all other kinds of viral proteins that block at different steps. So for example, PKR has to dimerize in order to phosphorylate. There are proteins that block that. This is amazing, and this is only a partial list. So apparently this is a very important defense mechanism, and every virus is involved with it. Another example of eIF2 phosphorylation is using another protein kinase called PERK. PERK stands for PKR-like ER transmembrane kinase. So you can see it was discovered after PKR was discovered. And this is a kinase that monitors the state of proteins in the endoplasmic reticulum. You know, proteins that are destined to be transported out of the cell or put on the surface go through the ER. And when proteins misfold in the ER, which they do if the cells are subjected to a variety of conditions like virus infection or hypoxia, toxins, and so forth, proteins begin to misfold in the ER. And the PERK system detects the misfolding, and it shuts down translation because it's thinking, well, it's not thinking. The idea is that if proteins are being misfolded, we don't want to make any more proteins until we get to the problem of the misfolding. So let's stop translation, sort out the misfolding issue, and then resume. So that's the idea here. And so virus infection triggers ER overload. A lot of viruses make a ton of glycoproteins and put them in the ER, and that triggers this system right away. So PERK is a ER transmembrane kinase. So it exists on the ER membrane. It has a part on the outside of the ER and a part in the lumen. Part in the lumen is joined with a protein called BIP. BIP stands for binding protein. BIP recognizes misfolded proteins. So when you go from folded to misfolded proteins in the ER, these bind BIP. They remove BIP from PERK, and then PERK dimerizes. It activates itself by phosphorylation, and then it shuts off translation by phosphorylating eIF2. So here it's not double-stranded RNA that's activating PKR. It's the misfolded proteins that are titrating away BIP from the ER. So another way that the cell tries to combat infection. When cells are under stress, they try and shut down translation, as I've just told you. So in a normal uninfected cell, you have perhaps circularized mRNAs with translation proceeding actively. Under conditions of stress, virus infection, toxins, nutrient deprivation, anything that will trigger the eIF2 phosphorylation pathway, you have what are called stalled 48S complexes. So these are mRNA ribosome complexes where translation has stopped. Because the cell thinks that they're in trouble and they better stop making proteins. These, very recently it's been found that these stalled complexes get assembled into very specific places in the cell, and they're called stress granules. And you can detect them. They're very obvious. They have specific proteins that comprise them, and some of them are shown here. So you can take antibodies to these proteins and look in infected cells and find these wonderful punctate stress granules in the cytoplasm. And within them are these stalled mRNA ribosome complexes. So the idea is that it's a storage place. Let's put these away until such time comes that we can resume translation again when conditions get normal. And you can show that, in fact, when you remove the stress from the cell, these dissolve, The mRNAs come out and resume translation. Very clever way of sequestering these mRNAs until such time that it's safe for them to be translated. So what do viruses do to antagonize it? A number of viruses have been shown to block the formation of stress granules. Why? They don't want their messenger RNA being sequestered in these nice punctate dots in the cytoplasm. They want to make protein. So West Nile virus, dengue virus, poliovirus, all encode G products that antagonize components of the stress granule and either prevent them from forming or take them apart once they're formed. Again, the virus wants to translate, and it doesn't want to be sequestered in these stress granules. So another very interesting way of interacting with the translation system. Now, the EIF4F is a target of activity. I mentioned this earlier. EIF4F is this whole complex, EIF4E, 4G, and 4A. 1, 2, 3 proteins. And a number of components of EIF4F are targeted, and I'm going to talk about just EIF4G today. And particularly the cleavage of this protein by two picornaviral proteinases called 2A and L. So there's a cleavage site on 4G for these proteinases, which essentially removes the binding site for EIF4E and blocks the ability of this to serve as a bridge between 4E and EIF3. And remember, the iris does not require a cap, so it doesn't care if there's no EIF4E interacting with 4G. So these viruses cleave here, and their irises work perfectly well in the absence of an intact EIF4G. So here's an experiment to show you what happens in, say, polio-infected cells. So if you look at different hours post-infection, you infect cells with polio, and then 1, 2, 3, 4, 5 hours post-infection, you measure the rate of protein synthesis. So what happens is, within an hour, the rate begins to decline. Between 2 and 3 hours, there's hardly any protein synthesis, and then it resumes again. And this latter protein synthesis, which peaks at 4 hours, is all virus-specific proteins. So the virus has shut down the host protein synthesis and replaced it with its own. And that's shown on the right on this polyacrylamide gel. At 0 time after infection, all the cell proteins are still translated. You just see a smear on this gel. And then slowly, at different hours post-infection, the host protein translation is inhibited and replaced by the synthesis of viral proteins. How does this happen? It happens by a cleavage of EIF4G. So here's another experiment where this is exactly on the upper left panel A, the same experiment that I just showed you. It's a time course of polio infection, and we see the shutdown of cellular translation replaced with viral translation, viral protein synthesis. And then panels B and C are Western blots looking at the state of EIF4G. It turns out that there are 2 isoforms of EIF4G in cells, 1 and 2. You can get an antibody against either one, so you can do a Western blot. And look what happens at the early times after polio infection. Here's intact EIF4G1. And then here at the 1 hour time point, you see some degradation. And eventually it's all degraded. Same with EIF4G2, except this happens later in infection. It starts at about 1 3/4 to 2 hours. So this is the one that actually correlates with the shutoff. This happens earlier than the shutoff occurs, but both are important. And what is happening when this protein is cleaved is that the N-terminus is being cut off. And it showed you in an earlier picture that little N-terminus cracked off. That's what's happening here. It's being cleaved by a viral protease. So there is a protease of poliovirus, foot-and-mouth disease virus, and other picornas that cleave right here at the N-terminus of 4G. And now 4E can't associate with it anymore, so it cannot bring, this complex cannot bring ribosomes to the cap. Because remember, 4E is the cap-binding protein. So normally you need to have 4E binding to 4G, as shown on the bottom here, so that the ribosome can be brought to the cap. But if you cleave here, that can't happen. And the iris doesn't care about caps, remember. The iris can have ribosomes bind internally, so it doesn't care, and that's why viral translation proceeds in cells where this cleavage occurs. There are also a number of other mechanisms of regulating translation. I want to point out to you, here's cleavage of EIF4G. The virus also induces dephosphorylation of EIF4E. Apparently, phosphorylation of 4E is important for recognition of the cap, so it's another way to decrease the affinity of 4E for the cap. And the virus also induces dephosphorylation of a protein called 4EBP1. And this protein, in its phosphorylated state, will induce the sequestration of EIF4E, such that cap-dependent translation cannot occur. Here's a nice structure of what's going on here. So this this 4EBP1 protein, this is a negative regulator of EIF4E. So this binds 4E and inhibits translation. It turns out that the reason it inhibits translation is because it binds the part of EIF4E that would normally be binding EIF4G. So that's kind of schematically drawn here. This little N-terminus of 4G has a depression in it, and that bit of 4E fits in there, but 4EBP1 is blocking it. So that has actually structurally been shown. On the top is the three-dimensional structure, high-resolution structure of EIF4E. Here's the cap moiety fitting into EIF4E. And on the back is where this 4E would be binding EIF4G. So here on this top, in part A, we see 4EBP1 binding, and then on the bottom is EIF4G binding. So 4EBP1 includes the part of 4E that would normally bind EIF4G. And finally, there's another way that a virus interferes with translation at the other end, if you will, by interfering with poly-A binding protein. Now remember, I told you there's good evidence that for efficient translation, an mRNA needs to be circularized. And it's circularized by this PABP poly-A EIF4G interaction. In cells affected with rotaviruses, which are real viruses with double-stranded RNA segmented genomes, host cell translation is inhibited. And the reason it's inhibited is because there is a viral protein called NSP3, shown here in green, which binds 4G and prevents PABP from binding. So it's very similar to 4EBP, except instead of binding and blocking 4E to 4G, it's now blocking poly-A binding protein binding to 4G. So by binding this viral protein to EIF4G, it excludes cellular messages, so they can no longer be efficiently translated, and they are replaced by viral messages. So here at the bottom of this picture, this is a viral message. The viral mRNAs are not polyadenylated, so they don't care about poly-A binding protein. Instead, they have a sequence, CCAGU, which specifically interacts with NSP3. So they basically can replace cellular mRNAs in this initiation complex in this way. So now they have a cap, so they bind nicely to 4E protein, and their 3' sequence binds NSP43, which has replaced PABP in this complex. So we think this is the mechanism by which these viruses inhibit cell translation. So all of these mechanisms have elucidated normal processes in the cell. So before we learned, for example, that the coronaviruses cleave EIF4G, we really didn't understand the role of that protein in translation. But because we had a probe that we could use to study the function of the protein, we understood what it was doing. So there are many examples of this, not only in translation, but hopefully keep this in mind as we go through different aspects of viral replication, and you'll see how studying virus-infected cells has really illuminated what happens in a cell.